Good. So what we arrived to after the marathon we did this morning was that we have these two views. So the Lamarckian view was that P of n given mu was something very big around the mean, while Darwin was something that looked like okay. So this distribution is a distribution that it's very broad. So if you plot it, it looks like something like this. Okay. So it has a very long tail, which means that there are rare but not too rare events where there are many mutants. Okay. So you can, instead of doing the calculation, you can simulate the process, and there is the code in the blah 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 lambda provided. And if you do that, so here it's the probability for the Darwin scenario, and the black points are like the events, while the red one is the Poisson, and you don't see anything like this. What you can see here is that there are events that appear like here, 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 here. Okay, that there was one event with above 6,000 mutant out of 10,000. Okay, so this is like what I did is running many simulations of this process. And so this event here is an event where the mutation occurred very, very early. Now, if you look at a distribution like this, you don't see anything. Okay, so in general, when you have distribution that are such long tail, it's better to plot them in a different way. Okay, and so one smart way to look at them is actually to look at what is called the cumulative distribution, which is basically the probability to have an m larger than a given m, which can be written as the sum from m from 1, u from 1 to m of p of u. So is the probability, the thing that you can plot is what is the probability that I see an event with more than some number of individuals, some number of mutants. Okay? And so if you do the calculation from this, what you get is that this probability should be equal, should be something that it's like 1 over n mu over n. Okay? And now, so you have something that scales as n mu over n. Okay? And when you have something like this, a smart way to plot it is to look at log p versus log n. Because if you plot it like this, it looks like a line. Okay? So if you replot the data in that way, the data, the simulations, what did I put? I think. No, but was here. Ah, there. Okay, if you plot the data like this, what you see is that now the data have this line here, and actually this line is exactly the one that corresponds to 1 over n, which is what you can do. Okay? And what you can immediately see is that if you have a Poisson distribution, there's a very different behavior. Right? Exactly because you don't have these rare events. Okay? So for instance, in this case, I think that the population was 10 to the 4th, the maximum. And you can see that you have 1% probability, right? Yeah. 1% probability to to see something which is more than a few hundred mutants. Okay? While with a Poisson, that never occurs, right? You never see anything more than 200. Okay? While here you have a fairly big probability to see something about 200. Okay? But this is the difference between the two predictions, right? And sort of this shows also that the simulation sort of agreed with the data. Now, of course, it's a bit different because we did like some assumption in the calculation, but it gives it. Okay? And so now, going back to the data, all right, so this is the experiment, is what we described. And the intuition here is what we said, right? So if you repeat the experiment multiple times, you have some events where the mutant appears early, and so then at the end you have many. And you have some events where the mutation doesn't appear, or some events where the mutation appears late. Okay? So you have a very broad distribution of the mutation. And so the point is that actually the sort of trick here, or the idea here, the very smart idea of Luria and Delbrück is that a signature in their experiment, a signature of Darwinian evolution is not in what is the expected number of mutations, but in the variability across experiments. Which, if you think about, if you come from an experimental background, it's very counterintuitive. I mean, suppose that you run an experiment. Right? Those who come from physics, you run an experiment, you think of any experiment you run in your physics course, or the one coming from biology, any experiment you do in biology. Right? If you replicate the experiment multiple times, what you typically want is that you get consistent results. Right? What they have is that actually a very strong feature of Darwinian selection, or Darwinian evolution, is that you have very, very different results across experiments. You have different results in a reproducible fashion, meaning that you know what is the distribution, okay? And you can replicate the distribution. But what you expect from this experiment is that when you count your plate, sometimes you don't see any mutant, and sometimes you see hundreds of mutants. Right? And so this is not because you are wrong as an experimentalist, but this is actually a feature of your system. Okay? And so these are actually the data of Luria experiments. Right? These are different experiments run with different conditions, and these are different replicates, right? And there is no need to plot anything to realize in which scenario we are. But this is clearly the Darwinian scenario, right? I mean, look at any line, you clearly see that you have very large events and very small events. I mean, think this line, you have 107, and then you have 0, 0, 6, 5, 5. This one you have 300, and the other are small numbers. Okay? So that you have this broad distribution that you cannot explain with Lamarckian evolution. Okay? Now, if you want to read something that is more modern, and is actually an application to real to more modern data, is this paper from some friends, also Maninos. And what they did is that they studied, they did an experiment not with bacteria, not with yeast. They did it with sort of model tumors. So you can have an organoid, which is like a sort of model of like a simple organ that is growing in vitro. Okay. And what you do is that you start with one cell, and then you have these things growing. Okay. You have these things growing, and then as it is growing, now the experiment does not last 24 hours, lasts 6 months, because human cells are very, very slow in growing. So mammalian cells. So you grow this clone, and then what you do is that you don't sequence. Okay? So what you do is that you sequence the genome of many of these cells, and then you count mutations. Right? And by counting mutations in different sites, you look at basically how old when the mutation arose. And the point is that now you have two technical problems which become very important. One problem is the problem of sampling. So you are not observing all the cells. You are observing a small fraction of the cells. So if something is very rare, you might not sample it. And you have a problem of sequencing errors, that when you sequence, you make errors. And so actually you have a finite window in which you can see the power law behavior that we discussed before, the decrease, but you can sort of see, okay? And what is cool is that then you can use this, so I sort of described it going quickly, but this probability that we described was mu times n over m squared. So if you know these quantities, you can calculate what is the mutation rate. And so what you can do is that you can do this experiment and sort of by multiplying this value by the frequency, you have an estimate of what is the mutation rate. Okay? And so you can use this experiment to estimate the mutation rate of this population group, okay? Which is pretty cool. So if you are interested in this, you can read the paper. Okay, the complete link is on the okay? Questions about this? Okay.
Did they say something or after?
So this peak here that you see is, I think, is the one given by, because here what you do is that you have also multiple sites in the genome. So this peak can be due to, I think, sequencing errors. That's the idea. But you have a model to explain. Then, I mean, if you look at the data very carefully, so you see that there are other deviations because you have mutation. The assumption here, one big assumption here, and this connects to the next point, is that mutations are neutral. What does it mean, neutral? What you're assuming here is that during this growth, we say dm t is equal to Rn, and we say that dn dt is equal to Rn. So that the mutants grow exactly as the resident population. In general, this is not true. When you have a mutation, it can be beneficial or deleterious. Right? So this makes things a little bit complicated. Okay? Other questions? We have to switch to the other presentation. But I
have a question.
Yes.
If we run the model like in a lot of iterations, I think my assumption is that as we increase the number of generations, that approximation of the rate is going to be worse because we have more variability between what are the last mutants based on the previous sensitive genes or not.
Yes, in the sense that the more, I mean, in general, there is a problem. I don't know if it depends on how long is the experiment, how big is the final size. The problem is that mutations are not neutral, so they have an effect. And so you have subclones that can grow faster or slower. I think that it's true that if you have a larger population at the end, so they grow for more, mutations that arose initially and have a different growth rate, you will see the effect, you will see a bigger effect. I don't know if this is
it was more like when we have like the parent and like the son. There is like a one mutation, but it is not like a really big difference between the original or like the parent and the son. But if you have like more generations, there will be a more distance, like genetically speaking. So like the rates of growth maybe will vary more. We have more and more generations because individuals are going to be more different as we increase.
Yes, in the sense that you have more. I mean, the difference in this model is driven by mutations. Right? So the reason why you have a bigger difference if you are more distant is because there are more mutations that make you different from the other cell. So this is what determines differences. a big assumption of what we did so far is that all mutations are equivalent. Right? In general, you can imagine that there is a distribution, right? That if you have a mutation, can be positive, can be negative, and so on. Okay. So, yeah. Okay. Other questions? Good. So, as I said, what we did so far is to assume that mutations don't affect fitness. They don't have any effect on growth. And now we want to exactly study when mutations have an effect on growth. So the effect of selection. Okay? So as we said in the introduction yesterday, selection is this force that makes that. So we have natural mechanisms in which individuals that reproduce more rapidly, that reproduce more, are more likely to produce more offspring in general. And so there is more individuals that carry those traits of the parents since the traits are hereditary. And this produces a force that changes the type of variation, the traits of a population. Okay. So what we want to study is exactly this force. Okay. So this is where we are in our plan. Okay, so this is a simple experiment where this is done in the favorite organism of money, so in yeast, cerevisia, which is the yeast that is used to make not all, but most of the beers. If you know the beer Ceres comes from this, and this is basically you track the frequency of mutations in an experiment, right, over many generations. So actually, the way these experiments are done is they are not done in the same way as, well, there is a commonality with the Luria Delbrück, but what you do is that you have your flask, you put in your organism, the yeast, this grows, then you take a small sample with yeast, you put it in new nutrient, they grow, right? And then you repeat this, right? You do it for 24 hours, every day for how much you want. We're gonna see. So in this case, I guess that you have 6 generations. If you dilute 1/2, yes, it usually yes, like it depends on. Yeah. So this is 100 days, so this is 3 months. Okay. So you do these experiments for 3 months. For 3 months, every day you go to the lab and you do this. Okay.
No holidays, no weekends.
This is what it takes to do experimental evolution. Now, this is then what you do is that at given points you sequence what you have, and each line represents. So you have DNA. Right? On DNA you have different letters. A, C, D, G, and you take one side of DNA, and what you look is you compare to the original cells that you have, and you ask how many cells on this particular site are different from the original cell, which is usually called the wild type. Okay, is it clear? So you take this point here in DNA, and you ask how many cells are different from the wild type. Okay? And so different lines represent different positions in DNA. And what you see is that you have this trajectory, right? And for instance, these black trajectories, what you see is that they go to 1, right? And so the idea is that these trajectories here are trajectories that are selected, okay? That are selected, and somehow you started from 0, and then you go to 1, okay? If you worry why here they are different, is also because here you make a big error on reactants. Then you see that these go to 1, so all the individuals at the end, they have 1. This is what sometimes is called a sweep, okay, genetic sweep. And what you see is that you have some mutations that were not present in the wild type, and then they go to fixation. So all the population is like that. And the idea is that this, when you see something like this is a sort of the outcome of selection. Okay? The reason why this is happening, it's not necessarily only the outcome of selection, but the idea is that this could be the outcome of selection. Okay? Because these individuals that carry this mutation have an advantage over the other cells. Okay? And so what we want to do in this part is to study this type of dynamics. Okay? Now, we talked about this experiment where you run this experiment for 3 months, right? And this seems very long. Now I want to go to the
real deal.
The real deal. Okay. So in 1989, when I was 2 years old, a guy started an experiment where he did this with Ecology. And every day, right, it was growing, transferred to a new medium, growing, etc. Right? And the experiment started when I was 2 years old, and it's actually still going on today. Okay? Okay? So they have like 12 populations, started from, so they repeat this for 12 populations in parallel, where they started from the same individual, so they're identical at the beginning, and they have this experimental evolution experiment, this evolution experiment going on in parallel for these 12 populations. Okay. Now, if you track, in this case, there are based on the dilution, there are like about 7 generations. So if you make the math, it's like now it's 70 over 75,000 generations. Okay. Now, if you want to compare, so with this plot of humans, so if a generations of humans is 20 years, this means 1.5 million years ago. So you have, comparing to humans with all the problems, you have an experiment that is going from around here. Okay? When there was there were some Homo, but were like, there was Homo erectus around. Okay? This is the kind of how back this was. Homo sapiens was very, very away from that. Okay? So you are really looking at an experiment over scales that are very, 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 very long. Okay? And then you do the plots. You can do the plots like the ones I showed you before. And this is the kind of plots you get. Right? This is for the first 60,000 generations. So these are populations. And what you see is like these lines that corresponds to, each line corresponds to a site in DNA. And what you see is that you have like all these lines going up, sometimes going up and down, up, 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 up. And these are all genetic sweeps. Okay? So where you have these things, right? And sometimes there is something much more complicated that is going on, which I'll describe later. So now what?
You know what media they use? Is just like
It's
whenever.
Is M9 plus glucose.
Yeah.
The simplest medium. They designed this to be the sort of, the original idea was to do, let's do evolution for a very long time. This is called the long-term evolution experiment. The idea was to do the most boring environment you could possibly imagine. Okay? So it's something which is like really, so M9, it means that you give the basic salt you need to grow, and glucose is the simplest source of energy that you can give. Okay? It's really the simplest environment you can imagine. Now, what is interesting is that, so you clearly see mutations, right? You clearly see mutations that the genetic structure of the population is changing, right? So here are different sites, and they are changing all the time. Okay? And this is going on, right? It doesn't stop. Now, these are not just, so this plot here is number of mutations, right? So you look at the number of mutations that you have. This is a cumulative, no. So this is how many mutations you have seen. And basically, you see that this grows in our populations, and you have this population that explode. Right? And this is because there are some populations where you have a mutation that changes the mutation rate. So basically, there are some populations where all the mechanisms of repair of DNA messed up. And so these cells start to mutate much more. And having an higher mutation rate in this simple environment gives them an advantage. And so you have these hypermutators that grow. This is the fact that mutations keep accumulating, right? Which is interesting. The other interesting thing is that this is fitness. How you evaluate fitness? You compare one individual taken from any point in time with the ancestor, and you see how differently they grow. And fitness is actually keep increasing. This you don't really see, but there is a big increase at the beginning, but this is always increasing. Okay? So fitness, how fast these things grow, is changing all the time. It's continuously changing, right? So there is selection that is acting and is making this organism to grow faster and grow better than the ancestor. Okay? The other interesting aspect is that if now, instead of focusing on all the mutation that appears, so all the mutation that are here, what you focus in is on these events that go to fixation. These events where you go up and you arrive to 1, and you ask how many mutations arrive to fixation, right? Basically sweep through the population and arrive to fixation. There is something which is also interesting, which is the fact that evolution looks like it's not continuous. You have nothing happens, then there is a bunch of mutations that arrive, then something is low, then nothing happens, then boom, nothing happens, boom, nothing happens, boom, right? So you have these phases of stasis, and then a lot of mutations sweeping the population at the same time. The other very interesting result is that in these populations that we were seeing here, right, you can sort of see here that it's like there are two groups of mutations that go along. And this is because evolution sort of created an ecology. Okay? So there are two clones, so there are two types of different cells, okay, that start to coexist. Okay? So you start from the same ancestor, but at a certain point, these two populations become different enough, okay, that they are like two different species in some sense, or ecological species. They behave differently, and they coexist. Okay? The reason is that there is one that grows slowly, that produces something that is needed for the faster growing, and this creates a feedback. And so they coexist. And you see that they coexist for a very, very long time. So this is like you start with Homo erectus, and at a certain point there is the Neanderthal and Homo sapiens that arise, and then they kept coexisting until today. Okay? Which is pretty cool. Okay. So now I know it's your favorite moment, in which I take this instrument of torture. So let's think together about what is the simplest model that we can write. Again, it's always about the simplest model. The simplest model that we can write. I mean, we describe mutations, and what we want to do now is to discuss what happens when a mutation arises, and this mutation has a different fitness. Okay? So let's say that we start, what is the simplest possible model of a population growing? Okay, so we started like this, right? Now I'll change notation. Instead of R, I will write F. Okay? This is a population where all the individuals within the population are the same. They are all equal to each other. Okay? Now let's imagine that they are not all equal to each other. There are different types within the population. Okay? There is, I don't know, type 1, type 2, let's say type N. Okay? And so I have Ni. Now, what I have to do if I have to assume that these individuals have different fitnesses, that there is a selection, that selection acts differently on them. What should I add here? What is missing? Suppose that these different types, these different clones, these different mutants grow differently. How can I capture this effect that they grow differently?
Another array.
What does it mean? Yes? What should I write?
A new pair. Another constant of array.
So, what do I do here? A function of what?
Time.
Okay, of time, it will mean that over time they grow differently. Right? What I want to say is that these different types are different from each other.
Maybe like a linear combination or not, like rates with their own number of like array times the individuals with a specific mutation.
Yes, this is what we have, right? We have the number of individuals with a specific mutation, and I'm looking at the growth of the individuals with a specific mutation. What I want to say is that individuals with that specific mutations grow differently from individuals with another mutation.
It's right.
Yes. Good thing it's just this. Okay. Okay, so I have this, right? And then suppose that I'm not interested in the total population now. I want to study these plots that we showed before. So what I'm interested is in the fraction of individuals with a given amount. So I say that I have a total number of individuals, which is sum over j and j. Okay. And what I care about is xi and I over n. Okay. So I want to write an equation for this. How do I do it? So what is this equal to? Come on, you know how to take derivatives. Even when you are tired. You have to take the derivative of this, right? So you have to take the derivative of n times 1 over n, and then ni times the derivative. Yes, just that. So how do? You have 1 over n d ni t plus ni d t 1 over n. Do you agree? Do you disagree? There are people that didn't do any action, so they didn't. Okay. So this quantity we know. This is fi ni over n. Okay. Now, what is the derivative of 1 over n? The derivative of 1 over n is minus 1 over n squared times the derivative of n. What is the derivative? Here, yes. And here I have to calculate the derivative of n. How do I get the derivative of n? I take the sum. derivative of n dt equal dt sum over j and j, which is equal to sum over j d and j dt, which is equal to sum over j fj and j, which is equal to n sum over j fj xj. So if I put everything together, what I obtain is xi fi minus sum over j fj xj. Now, you win, I don't know, fantastic prize if you tell me what is this quantity here. So xj is the fraction of individuals of type J. And F is their growth rate, or let's say their fitness. So what is this quantity? Another way to call this quantity.
The probability.
Not the probability. So what is the probability? It's related. What is the probability that if I take an individual at random is of type J? It's proportional to it's equal to the fraction of individuals of type J. So the probability that a random individual is of type J is xj. So what is that? Well, this is the average. Oh, didn't win the fantastic prize, so I keep it for me. So this is the average F. So it's the average value of fitness in the population. So what you get here, it's a very elegant equation, which is also called a replicator equation, which says that the fraction of individuals of type I, they grow proportionally to the difference of their fitness minus the average fitness of the population. Okay? So now this quantity here, of course, depends on the x. Right? But this is telling you that any given moment in, so this is a function of time. Okay? At any given moment in time, you have to ask, is this type more fit than the average? If yes, it grows. If not, it declines. Okay? Right? Makes sense. Okay? Now, if I start with a fixed number of mutants, okay, what do I expect that it happens at the end? And so I have fixed number of mutants. Let's say I have F1. I can order them by fitness. Okay. What is going to happen at the end? So you start with some average, and then the ones that are above the average, they grow. And the ones below the average decline. So what happens to the average?
Stays the same.
No, because you have more individuals above,
Yeah.
so it grows. So there will be more individuals that decline and fewer that grows, so the fitness will grow more. Now, if you have a population, what is the maximum value of the average F you can have? You can choose all the X such that the average is the maximum. The maximum you can have is when all the individuals are equal to the maximum fitness. Okay? So if you take this thing here, what it predicts is that at the end, so e going to infinity, xi is equal to 1 if fi is the maximum of the fitnesses. Okay? So this is the survival of the fittest, right? You have n different types, they have different fitness. This model predicts very simply that only the fittest survives. Right? So let's just to clarify this, let's look at what happens if we have just two types. Okay? So we have just two types. So I can take that equation. And so I have two types. And so I have these two types are type capital A and type small a. Okay? And let's say that the fraction of individuals of type capital A is x. The fraction of individuals of type small a is 1 minus x. Okay? X. A. Okay? And then this is thickness A. Okay? And now let's look at our equation. And let's focus on this one. Right? So what is this x dt? Well, it will be equal to x at A, A minus x times FA x plus FA 1 minus x. Okay? So if you put this together, what you get, and you do the algebra, you get FA minus FA x 1 minus x. Okay? That's what we get. Now, how do you solve this equation? Well, then this equation can be solved. You can solve it by parts, as usual. And so if you solve this equation and you assume that at the beginning you have some x0 individuals, what you have is that at time t, what you expect to have is x0, 1 minus x0 plus x0 e, let's call this s, e to the st, e to the st. Okay? So this s is the difference between how fast this mutant grows minus the other mutant. Okay? How fast the other grows. This quantity here is sometimes called selection coefficient. Okay? And so what we can see is that if s is larger than 0, what happens when the time is very large? If the time is very large, what here you have something e to the st. At the denominator, what do you have? Huh? Yes, this goes to 1, right? This term dominates the denominator, and these two cancel. Okay? And what happens if s is lower than 0? Yes. Goes to 0. Okay? Which is what we said earlier, right? So if capital A is more fit than small a, the frequency will go to 1. If it's smaller, it go to 0. Okay? Now, if you do you see if I write here or not? Yeah. So let's say that S is positive. This means that if you start from some point, the trajectory will look like this. Okay? This is called sometimes also the logistic curve. Okay? So we grow and we go here. Okay? While if S is negative, it doesn't go there. Okay? So this is what we expect sort of in the simplest possible scenario. What am I looking for? The okay. Now, this is what okay. Now, if you look at these trajectories and you compare these things, you find that there are some differences, right? So there is sometimes where it almost looks like it's the same, like for instance in this case. But then there are also other stuff that happens, right? So for instance, this goes up and then down, this goes up and then down. There are things that do something a little bit messy. Okay? And moreover, I mean, so we want to understand what's going on. Right? And if you go back to these curves, you can sort of see it even more, right? That you have like this transit and it's not clear what's happening. Okay? So the point is that in doing this calculation, we are neglecting something which is actually very important. Okay? We are neglecting something which is very, very important. And that's something that is very important is the fact that when you have one mutant, when there is a mutation, you have typically one mutant. You have one single individual. Okay? And so if you have a small population, if you have a small number of mutants, what do you think is the kind of process that can affect these results? Something we have seen. So when we talked about exponential growth, then we described about the fact that if you have a birth rate and death rate, and you have finite number of individuals, then stochasticity can play an important role, right? And so the point is that you can imagine that if you have one single mutant, okay, this single mutant, even if it grows faster than the others, is subject to these fluctuations. Right? If this single mutant dies before reproducing, the single mutant is gone. Okay, so we need to take this into account to study this kind of dynamics. Okay, and so the idea is so now one can study it in multiple ways, but let's say that we want to focus on a large population of individuals, and then we focus on some mutants N. Okay, and these mutants can grow, or these mutants go to extinction. Okay, and what I'm imagining now is that this population is somewhat fixed. Okay, so far we've seen only it's going exponential. The reality stuff are fixed. And so typically, if the mutant dies, there is something else that has, if the mutant grows, there is something else that has to die. Okay? So what we brought earlier is something like dxi with t equal to xi fi minus f. Okay. And so I can sort of, in a very naive simple approach, I can interpret this as something related to birth, and this is related to death. Okay? This is a very naive, stupid approximation. Okay? So here I have my mutant with some fitness FA, and I would say that this grows with, so this rate of growth is this one, and the rate of death is this one. Okay? This thing is done in a sort of, I'm doing this in a very simple approximation. Then one can do things more carefully, but the result is the same. I'll explain them more carefully later. So we have this simple process, right, where we can sort of map to what we have discussed so far. Now, the idea is that I'm imagining that this mutant type, has an initial population which is much smaller than the total. Okay? So if this is much smaller, this average fitness is basically the fitness of the other type. Okay? Smaller thing. So I have this process, right? And I have my mutant, and let's say that the mutant starts from 1. Okay? And then what I want to know, and this mutant has an advantage, so FA minus FA is positive. Okay? So on average, what I expect is that this grows. Right? But then I have stochasticity. So what happens in this scenario?
Some can go extinct.
Exactly. So I have some trajectories that go up, and I have some trajectories that go to extinction. How many we go to extinction? Well, we can go back to the notes and look at it. If we do that, what we discover is that the probability of going to extinction is equal to this thing here, the death rate, well, was the death rate over the birth rate to the initial population. So since when you have a mutant, you have 1, these things will be FA over FA. Okay? So the probability that you don't go to extinction, which I call fixation probability. The idea is that this grows and then basically this process take over and they go to fixation. So this probability here is equal to 1 minus FA over A, which I can write this way, and I can define it as S. Okay? What I discover is that the probability of fixation is S. What does this mean? This means that suppose that I have a mutation. So if we go back to the this point, this percentage here is exactly this S. Right? Now, this is calculated over many generations. So you see that over 10,000 generations, it increase of 20%. It's a big change. On a single mutation, what you expect that this number is very small. Okay? So this number is telling you if you have a beneficial mutation, how big is what is the effect of this beneficial mutation? Right? How much faster does a mutated offspring grow compared to the parent? Okay? These numbers are very small, typically 10 to the minus 3, let's say 6, depends on the organism, depends on the condition. But these numbers are small. Okay. Now this means that if you have a mutation, and this mutation has a positive effect, the probability for the typical positive effect, the probability that a mutation go to fixation is extremely small. Most of mutations that are beneficial don't go to fixation, they go to extinction. So it actually takes many beneficial mutations in order to have one that goes to fixation. Which is completely neglected from here. Now, if you want to play with the notebook, you can do a model which is a little bit more careful, that consider fixed population. This model is called Moral model, and you can read more about it. I don't want to spend more time on this, but you can simulate it, and basically, I have to erase this. And so this is the kind of things that you can ask to the model, and so this is the fixation probability. And this is the fixation probability as function of the population size for different values of S. And basically, for instance, if you look at this point here, what we predicted is 0.5, which is sort of here. Okay? So you have the lines, which are the analytical prediction, which is slightly better than what we predicted. You can do a little bit better. This is like a sort of naive estimate, which works for, well, anyway. But you can do a little bit better, and this is the prediction of the analytical prediction, and the points are simulations. When you run many simulations, when you have a mutant, and you check whether it goes to fixation. Okay? Something interesting. I don't want to spend, I don't have the time to spend time about it. But here there is also one case where you have deleterious mutations, so mutations that decrease your. And you can see that if the number of individuals is small enough, you have a probability that the deleterious mutation go to fixation. Okay? So you have the probability that a mutation that is worse, that is bad, actually spreads in the whole population. Okay? And this is extremely important. This kind of phenomenon is extremely important if you have small populations, for instance, endangered species, which keep mating each other, so there is not a pool of genetic diversity. This thing here is what happens. And actually, a recombination, so having sex, is one of the reasons why recombination is important. Okay? So there is an old story about this corner, which you can play around with them.